Ahora sí.
Hello, hello. ¿No? Hello, hello, hello.
Claro, sí.
Hello, hello. Hello. Hello, hello.
Vas a verlo un poco mejor.
Hello, hello. Pues no, yo me uso el micrófono.
No, ele vai subir um pouco mais.
Ah?
Férias?
Não, mas assim fica.
Bota no bolso.
Mas assim fica ruim.
Não, não, pode botar aí. Bota na lapela que ele vai subir um volume um pouco mais alto.
Aham.
Acho que melhor aqui. Te incomoda?
Não?
Cala.
Alô, alô? Não. Tem pointer? Andrea, tem pointer? Apontador? Para passar? Nossa.
Só que o pointer, olha só, olha para lá. Ele aponta aqui, mas na tela ele não sai. Mas dá para passar os slides.
Ah,
Peraí, deixa eu botar aqui para você.
mas como esse negócio aqui? É interessante, né?
Deixa eu ver, não passa. Botei aqui, ó. Ah, será que eu não vou botar? Peraí.
Não.
Eu vou pensar aqui.
Tinha que tirar. Não, não, pode tirar aqui. Hã? Não, não passa?
Como tá? Aí sim. Tá. Com esse para frente, com esse para trás.
Ok. Aham, então, pointer não tenho, né? Tá. Ok. Então. Acho que dá para fazer assim. Uhum.
Buenos días a todos. Disculpen el atraso. Tuvimos algunos problemas técnicos. Yo, después que termine el doctor Ulrich, nosotros vamos a conversar sobre mañana y pasado mañana, ¿ya? O quizás les digo inmediatamente, mañana es online, pero la asistencia es obligatoria. Es el único día que nosotros vamos a contar asistencia obligatoria por Zoom, ¿ya? Para que haya presencia y participación también. Esas charlas son súper importantes. Y el paseo el domingo es en el Cerro de San Cristóbal. Nosotros vamos a mandar por email la información de dónde nos encontramos y cómo vamos a proceder, ¿ya? El doctor Henning también va a hablar en inglés, al igual que el doctor Stevens. El profesor Henning Ulrich es profesor titular y el jefe de laboratorio de neurociencias en la Universidad de São Paulo. Está centralizada su investigación en la señalización purinérgica relacionada con neurogénesis, neuroinflamación y neurodegeneración. Él es un miembro fundador del Club de Purinas brasileño y fue vicepresidente y presidente de varios congresos relacionados y con otros tipos de investigación como bioquímica, etc. Él recibió el el premio de Fulbright Brasil Global Health Chair y es un editor asociado para la revista Cytometry, con especialización en citometría de flujo. Y el doctor Henning, además, funciona como asesor científico de Tissue Gnostics. Él va a mostrar varios resultados relacionados entonces a su utilización de este tipo de equipos en nuestro ámbito de ciencias. Y en la tarde tendremos en el curso de calcio también una actividad relacionada a analizar imágenes de este tipo. ¿Ok? Thank you very much, Ulrich, for coming. It's a pleasure to me to invite you to this meeting. And I hope we have a good presentation and that you make several questions for the doctor. Thank you very much.
So, thank you very much for Andrea for inviting me to come here. And it's a pleasure and an honor to talk to you about cytometry. Yes. And so it is great. I think it's a great activity what you're having here. And I hope that I can also contribute to this in the future. So I will talk today about imaging cytometry, yes, and how you can use this to study events of brain development and also what you can neurodegeneration. So what I need to say you, when I have this conflict of interest, when I'm a scientific advisor of tissue gnostics. And now, so tissue gnostics is building up some material outside. I just wanted to tell you a little bit what is about that. And first of all, what we are doing, so I know that this may be very simple for many of you, what I tell you, but I heard that there are also high school students here. And what I want just to show you in the beginning is how do you study cells? What is cytometry? Cytometry is that you use fluorescence or other type of staining, and then you stain cells for epitopes. What is on the on the surface of the cells, and what is also, you can also label intracellular epitopes, yes? And normally you do this by fluorescence compound, which are coupled to antibodies, and then if it's antibody bind, then you get fluorescence, okay? This is very simple. But now, so there are two types of labeling that you can do. You can get a fluorescence labeled antibody, which you incubate directly with your target cells, and then you get, if it binds, the fluorescence signal, yes? Or what is done more frequently, and it's better because it enhances the signal, and it's also more specific, but you use an indirect recognition. You have a primary antibody, which has no fluorescence marker, and against this primary antibody, when it's bound, you direct a secondary antibody, which binds on top, and gets the signal. And like this, it's possible that various secondary antibodies are binding. And now, so if I tell you cytometry, oh, you will think about flow cytometry, yes? Because flow cytometry is a technique which is very much used in clinics and in research. And in flow cytometry, actually what you do, you take cells out of your culture dish, yes? You need to take them out with trypsin, you need to get them into suspension, you need to separate them mechanically, and then you send them through, what did I do? No, it's okay. No. So then you send them through to a flow, yes, and then you see there's a laser, and this laser detects, excites fluorescence, and the fluorescence emission is detected, and then basically captured by an analysis workstation, okay? And this basically each of this, each dot that you see here is correspondent to one cell. Yes, so it means that the cells are separated. They flow through. They basically flow through the laser detection. Yes, and if there's a fluorescence signal, then each of these points is a cell. Yes, like this, because why are you using flow cytometry? Because you get a very quantitative analysis. Yes, but there's a big problem. But flow cytometry is good for cells which are in suspension, like blood cells, lymphocytes. Yes, and this what you have in the plasma. But if you want to analyze tissues or cell cultures, then actually there are better techniques. And what I will present you here is a technique which gives you all this type of information, but plus the single cell information. You see the cells in their natural environment, and you see the cellular context. And you don't destroy the epitopes, because if you remove cells from the dish by trypsin, you will destroy the extracellular epitopes. Let's see. Okay. So now, what I want to tell you is that you basically, you see here the difference, what I put here as a fluorescence imaging microscope, what the tissue agnostic system in principle is. And then you see on down. So I won't go through all this type of stuff here. But there is one thing that is very important, that in former times, there was used basically a human who was counting positive cells, 200 cells per slide, 300, and it's a very time-consuming work, yes, and it's not very objective, because I can say this cell is positive or not, while the flow cytometer analyzes the cells as dot plots. What we will do now is to learn how we can transform images into segmentation and do a quantitative analysis. And for this, we use the TissueFac system. And what you see here, you see this is a microscope. It is an inverted or upright microscope, depending on what your applications are. And you see it has a table which is automated. So it's a size microscope, but it is modified. And this table does an automated scanning, and then it puts together all the regions which are scanned, which are entire slides, and mounts a virtual image. Okay, and then you have all this analysis workstation. And here, so this is just where you see a very simple example. Yes, what I told you before, that you can for example, you will count here Ka67 positive cells. For example, these are cells which are going through into mitosis. Yes, and then you see in this case by immunohistochemistry. Yes, you have either blue or brown cells. Yes, so the brown cells should be the positive cells. And now, if you give this to a human observer and say how much of these cells are positive, then you get very much different opinions and counts. And this has been done. It has been done between 1 and 40%. So which is a very big difference. But now we are using a segmentation program to study this accurately. And this I'll show you in the next slide. So it was between 1 to 40% by human observation. And now what we are doing But we use an automated analysis, and we say, in this slide here, there are 14.5 cells positive for the staining, which is much better than the human estimation between 1 and 40%. Okay? And how do you do this? Because of this, I showed you in the beginning the flow cytometry. We do the same here. But the only thing, we detect cell per cell in the dish. Yes? Yeah. And then you establish here, but in the absence of the secondary antibody, what is the autofluorescence or the autostaining. And when you see that you have, you can establish a background, and every point on top of this is a positive cell. Okay? So now, how can you do this? So what we do is actually, you see here, a tissue infiltrating by leukocytes. And we stain here for 3 markers. For CD4, CD8, FOXP3, and we use DAPI. Why you use DAPI? You always, you need to make a staining of the nucleus. Yes? And the system will use this DAPI as a master channel for detecting the cells. Okay? So this system has now been very largely improved using artificial intelligence. Yes? But we'll see here, you see the segmentation, the automatic segmentation of the cells, which is here pretty good. Okay? And then you have, we go for the immune labeling. Yes? And now, for example, I will come to this later on. So you have, for example, here you have a normal skin, and you have a UV irradiated skin. Which is, and then, so if you irradiate skin, then you get an upregulation of keratin 17, and what you see with normal skin, and then you see the UV irradiated skin. And you can believe me, to analyze this just by observation, counting positive cells, is impossible, because you don't have a real definition here what is positive. And therefore, we can go again, we go here to analysis, and as you see here, that we basically, we set up in the norm, a control, what is positive, what is negative, using the absence of antibody labeling. Yes? And basically, in the not irradiated skin, you have 16% of positive cells, while after irradiating, you go to 45%. Okay? So this shows you very clearly how you can analyze this. And now, in this case, you have 2 things. You have an intensity, basically, of DNA staining, what you can do together, and plotted also the intensity of the antigen, which is the keratin 17 staining. Okay. So now, how do you do this? First, you do a detection of cell nucleus and cells, which is used as a master staining, always by using DAPI, or a DNA stain. And then you go for a second step, where you did a definition of the cell body, and you use an antigen measure mask. So in the afternoon, Bogdan, who is a specialist of tissue gnostics, will explain you a little bit better how this does work. Okay? And now coming back to the slide that I showed you before. And you see, coming back, you have a labeling of 3 antigens, not only of 1. One is the simplest thing. You have DAPI and you have a second channel. But using the system, how it has been developed today, depending on the configuration, you can even measure 20 colors, 20 antigens at the same time, if you would like to do that. And what you see here is that you have the DAPI labeling of your cells in blue. Yes? And then you see here, you have CD4, you have CD8, and you have FOXP3 labeling. But what can we do with this data? And how do we go ahead? In the next slide, you see that we can do the following. We gate the cells for CD4. And what you see here on CD4, that you have a background labeling, and all of what is on top of then you put into a gate, yes? And then if you click on any of these dots, it will show you the correspondent cell which has been labeled, okay? Also, if you click on the gate, it will show you all the cells which have been gated. So this is a large difference to flow cytometry, because it just shows you the power of the system. You have all the single cell information together with quantitative data. So now the same thing, when you label now for CD8, so the cells which are positive will show up here in red, and we put again here the gate, and now we do the same for CD4 and FOXP3, so all combinations are possible for you to analyze this, okay? Okay, so this was just a very short introduction, yes? But as I understand, you will have also outside a video who shows you shows you how the system works. So we recognize cells, yes, by DAPI staining, and then we combine this with antigen staining by an antibody or whatever what it be, and then we do a quantification, yes, and basically, but we can do more. So now I want to talk a little bit about my research projects. So first of all, neural projections. So I won't go very much in details with you, but one of my research lines is to study neurogenesis. And for this, we isolate neural stem cells from the embryonic telencephalon of a mouse, of a rat, take the cells into a culture, yes? And all this, what you see here, labeled what you see as a neurospheres, is a clonal expansion of a single neural stem cells. Yeah? And what you see on the right side is that you form neurospheres in the presence of growth factors, and then you remove the growth factors, and then you have the differentiation into neuron, astrocytes, and into oligodendrocytes. Okay? So now, this, of course, is a nice system to study in a very simplified form brain development or neurodevelopmental processes. Because if you look at here, these neurospheres, here on the right side, so they're undifferentiated cells, and this is stained for the cell nucleus, they just migrate out, such as is being done in cortex development by for the formation of cortical layers, okay? And then you can see that this is an excellent system, so now they grow out, they become neurons, yes? And we show neurites and so on. This is a staining for beta-3 tubulin, and beta-3 tubulin is a marker of young neurons. And in the next one, you see we can do the same for glial cells, for astrocytes, labeling by GFAP. And finally, you see you have a very nice system. And this system we can analyze by tissue cytometry. So now, how can we do this? Because I told you there's one parameter, that is the quantification of labeling, but we can do more. We can also, as you see here, you see basically, you see neurites, which are, for example, with a certain size, which are labeled here in blue. Yes, and you see here in blue. And there are others which are in red, which are more distant. Yes, and you can use the tissue FACS system to define them. Yes, and even say you have a length of whatever And you have of shorter ones, you have how many percent, of longer ones, how many percent, okay? So it means that you not only can analyze the staining, you can also analyze the morphology of these neurons. So now I wanted to tell you a little bit a work that we have done, and we have also published in very good journals. So my laboratory has discovered that bradykinin, which is a vasoactive peptide that everybody in Brazil knows, because it has been used for developing captopril. So bradykinin is an antihypertensive peptide, but it has also other factors. And what was shown in my laboratory, what I have shown, that it induces neurogenesis. So very shortly, so that you have two types of receptors, a bradykinin B2 and a bradykinin B1 receptor. The B2 receptor is around in many cells, including neurons, or differentiating neural stem cells, while the B1 receptor is a receptor which is induced upon inflammation. Yes, but both of them, they induce intracellular calcium signaling, and finally, with intracellular calcium signaling will lead to advance of neurogenesis. And what has been published, and so it's now more than 10 years ago by FaPespen, so this is the Sao Paulo Research Foundation, so we like this very much. And that basically, bradykinin would bind to a bradykinin receptor on neural stem cells, and this bradykinin receptor induces calcium signaling in a cell, and finally, this increases the migration of neural progenitor cells, and also it will lead to an increase of the number of neurons and a decrease of number of astrocytes. And also, what could show is, you see, I will just go on the image below. So we have three markers. DAPI is for the cell nucleus, beta-3 tubulin for the neurons, and GFAP is for the astrocytes. And on the right side, you see, if you treat them with bradykinin, yes, then you have almost only neurons. While if you inhibit bradykinin receptor by 140, there are almost no neurons, where you have the glial cells, okay? And so, means bradykinin induces neurogenesis. If you inhibit the receptor, then you have an increase in gliogenesis. And also, what is shown, that is the same thing, but we have also reduced migration and so on, if we look at neurospheres from a bradykinin receptor knockout mouse. And you see also that all this organized migration, what you see in the control, it's in the neurospheres from the knockout mouse is totally disturbed. So now, this is all possible to be analyzed by tissue cytometry, yes? But we used also tissue cytometry to go much further. What you see here is that bradykinin basically, as a mechanism what we studied, that it inhibits proliferation. It does not really inhibit proliferation, but it slows it down, because it induces an asymmetric division, where you have one cell which is more differentiated, becomes a neuron, while the other cell that is generated is undifferentiated. And it is an increase in the G1 phase. And then, how bradykinin is doing this? It is doing through activating an intracellular Magna signaling, and finally, this increases the number of neurons, okay? So now, how coming back to tissue cytometry? So now we wanted to show this is true in vivo. So we started with in postnatal rats, where basically bradykinin or its inhibitor were injected into the ventral gyrus of the hippocampus, and then we wanted to look about neural stem cell proliferation and differentiation. And the other compound that you see here is U0126 is an inhibitor of ERK. So the idea is bradykinin for ERK activates neurogenesis. And when you see now, what can you measure to study this? You can measure Ka67, because the cells will go into a cell cycle. They need to do this, to do an asymmetric division, to create a larger amount of neurons, okay? And when you see here, these are very few cells. There's a very rare events, because with stem cells, post-natal, are very few. And we are able to quantify with very few cells using tissue cytometry, yes? So we don't use any flow cytometry, we just do the section, and then we go for Ki67 positive cells, okay? And as expected, 140 inhibited the Ki67 staining, as it also did 140 the antagonist inhibited, as also it was inhibited by the blocker of ERK. Okay, so now we see this here. So that actually that we have an increase here. So a very low increase. If you look here at the two images, maybe by visual counting, say it's all the same. No, but if you are counting 100,000 of cells by using segmentation analysis, then you can make here the difference. I should show this here for Ki67, but you can also show the same thing for MAP2 and a neuron specific analysis. Okay, but now there's another point. Is it possible? There are two hypotheses. One of them would be that bradykinin would direct differentiation. And the second hypothesis, which is possible, that bradykinin would inhibit the proliferation of one of the subpopulations. For example, of neuroblast or of astrocytes, and by this you have more presence of one or the other cell type. So this is also something what we study. So you have a spontaneous proliferation and differentiation, or you have an inhibition of proliferation. And now, so the bradykinin-induced inhibition of global proliferation rate would be the consequence of neurogenesis, since neurons do not proliferate. Or bradykinin inhibition of proliferation would be the cause of neuronal enrichment that I am proposing here. And now, how can we do this, and how can we analyze this by tissue cytometry? So now, we use EDU staining, yes, which integrates into the DNA, but it does not denature the cells. The cells are okay. And then you come with a second antibody. Now, for example, you're staining for GFAP, for glial cells. And then you have the population of glial cells, and you have the cells which are positive for EDU. And then we can count this by tissue cytometry, and then we can see there is no change in proliferation. That's between the control and treated with bradykinin. And the same is true if we go now for nestin. Nestin is a neural stem cell marker, a neuroprogenitor cell marker. No, bradykinin does not alter the proliferation of these cell types. And it does not alter the proliferation of neuroblast becoming new young neurons. So that we can say, but to conclusion, that bradykinin induces neurogenesis. Yes? And it does not affect the proliferation of any of the populations that is in the dish. So, and the inhibition of global proliferation rate is a consequence of bradykinin-induced neurogenesis. You see, you can use this technique. It's extremely powerful. I'm not saying this because I'm an advisor of a company, but with this technique, so I have published papers in high impact factor journals. Okay. Yeah. So this work reveals a methodological approach for studying enrichment of defined phenotypes. So now, what we have shown basically that in vitro, in vivo, that bradykinin induces neural stem cells to become neuron. Inhibition of proliferation, induction of migration, phenotype determination. I did not show you this, going for cholinergic neurons, for example, that's which are enriched in the presence of bradykinin, and it has also mobilization of stem cells in vivo. So the question is about applications in therapeutic diseases. So now I come to a second point here, so that Andrea will like, because it has to do with amyloid plaques and neuroinflammation in Alzheimer's disease APP-PS1 model. First of all, I have time, yes? First of all, neuroinflammation is the principal trigger of Alzheimer's disease, yes? And what we think is that we have a dyshomeostasis when we become elder. And the inflammation basically persists in the adult brain. And one of a focus for doing inflammation are amyloid plaques. So which is something what should not be there. Yes? And it's being basically attacked by the innate immune system. Okay? So now, the idea to study this, and looking just technically at this, is to use, we can even inject beta amyloid into the brain, what I will show you after this, or what you can also do is using a transgenic mouse model, yes? And when you see here, but we have basically with APP-PS1, so this is a double mutation, yes? But you have amyloid beta deposit and memory loss from 6 months of age. And then you can, I don't know why, you can use this mice, you slice the brains, and then you, as you see there on the top, you stain them for amyloid beta, for astrocytes, and for microglial cells. Okay? Now, okay. For example, if we now look for amyloid beta plaques in the hippocampus. In this case, you use an antibody against beta amyloid, you stain, and then you detect using tissue cytometry. Yes? And of course, what is expected, that you have a lot of these dots in the APP-PS1 hippocampus, and you don't have almost any of them in the control. And there comes also in the artificial intelligence, which defines what you see in the control as a background. And it just zeroes it out. So it's very clear. So to control mice, we don't have amyloid beta. And you say to the system, so this very low labeling is background. Yes? And then it will just detect you the APP-PS1 labeling. So now, it's another paper where we published, where we were looking actually in plaques, and we transplanted neural stem cells and mesenchymal stem cells into the hippocampus. What would you expect by mesenchymal stem cell transplantation? Where normally they should be slow down the inflammation. So it would be reducing immune response. While also, you see, when you also stem cells, we have also transplantation, has the same effect somehow. And what you see, that you have on the, that you have less plaque formation, yes? And you see this number of positive plaques, the total area where you have a plaque do not change, yes? But this is something else. But you can see here that you very nicely can do this quantification of plaques using tissue cytometry, yes? So if you have questions, just you can ask, no? So now, using this animal model, we studied senescence, yes, and of microglial cells. So microglial cells are the resident immune cells of the brain. They are being activated by maybe by events. So first you have a systemic inflammation, and then it becomes damaged the blood-brain barrier. So the peripheral immune cells, they go into the brain. They're able to do this. But the main effect is when they induce the microglial to a pro-inflammatory type. And what you see here, that you have the microglial cells during sorry, I don't know. Yeah, what is microglial cells when they become adult? You see, they come from around. They have a lot of projections, yes, and then basically they go into senescence at some stage. They become aged microglial cells, and this means so that with projections get disconnected. And also, microglial cells, they have a function in the brain. And also, in Alzheimer's disease, they become senescence. And they are not anymore able to do phagocytosis of beta amyloid and to eliminate the beta amyloid. Okay? So now, so it means that we have microglial fragmentation in the cerebral cortex of Alzheimer's disease patients. And in the animal model, we see the same. So for example, so this was done with the APP PS1 cortex sliced, and when we stained for IBA1. So this is a marker of the pro-inflammatory microglial phenotype. Okay? And then you see here an original image. And this we subjected to tissue cytometry, to tissue gnostics analysis. So let's just look what we do. You see here, we identify the cell borders. Yellow, and then with the filaments, they are in green. And then the next slide is. So as you see here, you see exactly what we want to see. We have distant filaments, which are there in orange. So we have short attached filaments, and we have long attached. So the distant filament is the microglial cells which are in senescence, because they are fragmented. And we can define the size of these guys. And like this, we can analyze microglial senescence. So, first of all, then, just to go back here, we can also do a backward connection on short attached filaments, yes, what you will see in red. Then we can do a back connection, yes, on long attached filaments, what you see there in blue. Okay, and finally, what we can do is do a backward connection on distant filaments. Okay, so now, but this is not all of what we can do. Then also we can, of course, quantify microglial activation by tissue cytometry. So first of all, on the top, we see the automated analysis of recognition of cells in cell culture. Yes, and then this is cell culture. After doing the DAPI labeling, we do the IBA1 detection. Yes, and like this, we can see that we have basically a quantification. You see here on the left side, DAPI, you have this nuclear detection, and then you see the IBA1 Alexa fluor mean intensity, yes, and then you can say, okay, this is the number of cells which have a high mean intensity, and how is this related to the size of the cells? Okay, because I told you in the beginning that there's also a morphological process inside of that. They become round and they become large with a lot of filaments. Yes, and then we have, of course, a much bigger size. But it's possible to distinguish the amyloid ramified from round microglial cells. Okay, so this automatic analysis gives you a lot of possibilities to analyze. So now, but we have a number of IBA1 positive cells, the percentage of IBA1 positive cells. In this case, we injected the help of Sergio Ferreira, who gave us the beta amyloid into the brain of the animals, and we see here that we have an increase in inflammation. So this is not a transgenic animal, so this is an injected, and that we have an increase in the number of round IBA1 positive cells. So now, the idea is that maybe neural stem cell loss during aging and Alzheimer's disease affects microglial proliferation and activation. So maybe, you could think that endogenous neural stem cells may regulate microglial biology. Okay, if you inject them, then you may have the idea that you have an alteration in the activation. Yes, okay. So now, what we see, basically, we are looking again, injecting mesenchymal stem cells, neural stem cells into the hippocampus of the Alzheimer model and compared to a control. And what we see here, and this is exactly in agreement what we saw here before. If we go, that you have an increase in the number of IBA1 positive cells. So that would be even positive, because these cells are able of doing phagocytosis of beta amyloid. What you see here in both of these cell types injection. Because one problem of Alzheimer's disease is the absence of phagocytosis and of active microglial cells. Okay? And so, what we have learned from tissue gnostics, But this is an excellent approach for monitoring pathological processes in the brain, such as microglial morphology activation and fibril deposition. Yes? So I hope that besides some maybe a complicated part on microglials, which is very important in every neurodegenerative disease, I've shown you how you can use this technique to quantify. And so this is a technique of the future, because nowadays we will do not need actually to develop very much with segmentation, what Bogdan will show you. The idea is using artificial intelligence to detect this. So you need to feed basically the program with your negative control and what is considered positive. Yes? No, let's move it. And now, so just, okay, do you have 5 minutes? 10? Yeah? No, it's not. So the idea, I go very fast through this. It's not only that what you can do. Like this, we are looking for neurodegeneration, and just very quickly, it's an actual work, but I'm looking on pruinergic signaling. Pruinergic signaling are receptors activated by ATP. So it would be, for example, P2X, which are ion channels, and P2Y are G protein coupled receptors. So very shortly. Okay? But the idea is again the problem of inflammation, and pruinergic receptors are involved in inflammation in Parkinson's disease. So there's one receptor that we are looking at. It's called P2X7, and this is the cell death receptor. So it's in microglial cells, immune cells, and if it hyperactivated, it forms a pore, and with microglial cells it releases all types of pro-inflammatory cytokines using cell death. Okay. Now, the idea is also that the P2X7 receptor is a receptor which inhibits neurogenesis. And what we propose here in this case, in the striatum and substance nigra, that we may have endogenous neurogenesis to regenerate dopaminergic neurons. And this we see that is based on a work of Jonas Friesen, showed that this neurogenesis exists in the striatum of humans. And very fast. So that we inject to create the animal model on one side of the brain 6-hydroxydopamine. It's a neurotoxin, and it kills the dopaminergic neurons. Okay? And then, how do we measure this? There's a behavioral test, because after 1 week, the hemisphere is dead. So with dopaminergic neurons. And if we now give the animals apomorphine, which stimulates them, they start to run. But they cannot, because one side is dead. So they rotate. They do rotation. And we use this to look at the P2X7 receptor, brilliant blue G, so which goes into the brain, so we can just inject it intraperitoneally, and it goes into the brain. And now, very, very fast, tissue cytometry. You can use tissue cytometry to quantify. Now, what you see here is on the upper side, you see the control hemisphere, which has not been injected with anything. On the downer panel, you see the lesioned animal. So there is you stain with neurons for tyrosine hydroxylase, which is specific for dopaminergic neurons. And then you see, if you increase the concentration of brilliant blue G of BBG when these neurons become regenerated. Okay, yes, and this is in the stratum, in the substantia nigra, is the same. Okay, and now we analyze this by tissue cytometry, and you see here a quantitative analysis where we came. Okay, so conclusion is we have used tissue cytometry technology for assessing neuroregenerative effects of P2X7 receptor inhibition. So it was injected after 1 week. First, we gave 6-hydroxydopamine. After 1 week, we give BBG. And now just image cytometry is more adequate for analyzing cells in the environment as cell cultures or tissue sections without we need to collect them and separate them mechanically. Because of that, you don't want to use flow cytometry. Everybody, I will dissociate the tissue and I put it in a flow cytometer. So this is not the best thing. So this technique here is much more powerful for cells which are in a tissue or cells attached to the culture plate. Okay? And quantitative images of cells in their cellular context are available together with quantitative data. Okay? And just so this is about my group, and I'm also acknowledging here tissue gnostics, yes. And I was just the last. And the Institute of Chemistry has been received, actually it was my research, the premium for being a tissue gnostic reference center, because the company is present worldwide, and they give a premium to the centers who have published the most and the most high impact factor paper, in this area. So I published this is basically also in journals like Molecular Psychiatry with such an impact factor of 15 and this type of things. And if you look here down, we are together with the Reagan Institute of Boston at the MIT and Harvard. And obviously, since the company of Rupert Eckert is in Austria, we have also a very strong center of imaging cytometry in Austria. Okay? And so this just shows here when the prize was given to us. So, and thank you very much for attention. And I'm here to answer questions of you, yes. And I would really recommend to you today with Bogdan to learn, because I did not show you how you make this analysis. And since you are from different countries, so maybe as students, you may recommend this to your supervisors. Because there's another thing what I wanted to say. The system what I have is a confocal. So it is a confocal microscope. It's a spinning disk, and it has also an incubator with CO2 and oxygen, where you can do lifestyle imaging. So you put the dish there, and you tell the system when you want to image. And they are in the normal culture conditions, yes. And if you use dyes for vital cells, you can also measure for a long time calcium oscillations if you want. And then you get this very quantitative data. So this I just wanted to tell you.
Thank you very much, Henning, for this wonderful presentation. Questions? Christian.
Very nice technique. I have a question about, in the case of the bradykinin, when you do in vitro studies, like the cell culture studies,
Yes.
could you use tissue cytometry to follow the kinetics of the system? For example, the proliferation, and then you can, and now you mentioned about the calcium. Can you have kinetics of the different cells to see how the calcium change on time?
Yes, you could do this in a small region, yes, no? Because you cannot do it on the whole slide, because because the system, depending what kind of magnification you use, no? You will take time to scan. Yes, so in this case, no, if you want to go for calcium, of oscillations, then you would take a very small area, no? No? In this case, but all type of other markers, no, proliferation and so on, in living cells you can follow. Yes, no, no. The only thing is that you need to use vital dyes, no? So labeling, no? You cannot fix the cells, no? In this case, no? Yes, which leaves the cells alive, no? No? But for example, what you can do, no? You can use the tissue cytometry system for measuring cell cycle. Yes, as I showed you, EDU labeling or for example, yes, no? So which stains the cells, and you get an idea of the proliferation. Yes, no? Yes.
But you have labeled the protein like the GFP or something in order to get in instead
Of course.
Yeah, yeah.
That you can do anytime. Yes. Yes.
Thank you.
Mhm.
Questions? I have one, Henning, about a microglia,
Mhm.
because you said that you stained it with IBA,
Mhm.
right? And then you follow the inflammatory phenotype
Yes.
only because of the morphology. Is it okay, or do we have any other marker to differentiation M1 to M2
Of course,
microglia?
no, yes, of course, no. So you can measure, no? Of course, you're for M1 or M2 markers, no? What we did not do in this study, of course, yes, no. So this is just open, no, no. What you want to do, no? You can start a label, I think, for INOS, for example, yes, no. That would be pro-inflammatory marker, no? For example, no? You can label for anti-inflammatory markers, of course, yes, no? Yes. So the system, no, is not limited, no, in this, no? So my systems are those 8 colors, no? No? But there is basically the idea, no, that you can also use a deconvolution system, yes? So where you have like a monochromator, and you send all type of wavelengths, yes? And then you can differentiate between single wavelengths or single in nanometers, no? And then you use a tissue agnostic program that will deconvolute this. And like this, so you can measure maybe 20, you can maybe even measure 40 different stainings, yes? And then it becomes extremely complex, no? And what tissue agnostics offers you is also a very good program to do that and to analyze this, yes? Please.
I have just like a general comment, more a question regarding the specificity of some of the fluorescent dyes that we use to label nuclei. And for instance, I have recently noticed that DAPI can also stain amyloid in tissue. And this is something that people don't talk about. So I would like to know whether you have experienced that because you work with beta and alpha-synuclein aggregates, and whether you notice this kind of awkward staining, DAPI staining in the cells.
Okay, so this is interesting. Maybe so. This is the staining, what I've shown you in the control. There is maybe some staining that we define as unspecific. And when it was just gated out, yes? No? So when we say, okay, we go, we tell the system, so this may be unspecific, and then go for a solution, which will remove it. But it's a very good idea. So, and thank you for this comment. Yes?
Thank you very much, Henning. We don't have more time for questions here, but we can make questions outside in the coffee, either in Spanish, Portuguese, or English. Henning can take in the questions, and I invite you to visit the stand for tissue diagnostics outside, because they have some nice images also to show you, okay?
Let's just have a look at these images, yes, so, no?
Yes.
So I showed you something, but where's, yeah.
Thank you very much.
Gostou?
Foi a melhor que eu vi.
Hum?
Essa foi a melhor que eu vi. Das suas, essa que mais gostei.
Yeah,
É.
no?
Aham.
Thank you so much.
Mhm.
Amazing job.
Thank you, no? Yeah, that was, but we will talk